Hello students. So let us continue with the topic on the kidney that we were learning, and let us do in detail about the study of the nephron. The nephron is also called as the uriniferous tubule, and the nephron is made up of, the first part is called as the Malpighian body, which is itself made up of two parts. One is called as Bowman's capsule, and within the Bowman's capsule is a knot of capillaries, a bunch of capillaries it is called as glomerulus. Let us look at the diagram of the Bowman's capsule. So this is a cup-shaped structure. And in this cup-shaped structure, there are cells over here all around the edges of this cup-shaped structure. So either you can imagine this to be a cup, or you can imagine that this is a kind of a ball in which somebody has compressed a hand or a fist into it. So then this will give us this double walled structure. So these are two walls over here. So the Bowman's capsule is double walled. And these layer of cells that you are seeing over here, this is squamous epithelium. So it contains one layer of squamous epithelium, and the second layer is a visceral epithelium, but this visceral epithelium is made up of cells called as podocytes. So podocytes is visceral epithelium. Now these podocytes look like this, with several such digital extensions. So this is somewhat like a podocyte. There is a nucleus over here, but on one side, on this side, there are digital extensions. These are called as pedicels. So this is a pedicel. So all these are pedicels. Now what does a podocyte do is that the podocyte completely envelops capillaries in the glomerulus. So let us just see how the capillaries of the glomerulus will look like. So here, what did we say yesterday was that a branch of the renal afferent arteriole makes its entry over here, and now it capillarizes. That means it splits into several branches like this. So it branches several times into capillaries. I am redrawing one of the capillaries, let us say over here. So this is being shown as a capillary. Now this capillary has got several openings. All capillaries throughout the body have got openings. Only thing is that here the openings are a little larger. These openings are called as, or they are like windows, and hence these openings are called as, a single one is a fenestra, and all these are called as fenestrations. Fenestra, which means window. So these are openings everywhere. Any capillary throughout the body has got such openings through which white blood cells can move out. Similarly, plasma can move out. So there are openings are always there, but only in the glomerulus these openings are slightly larger, so as to allow the plasma and quite a lot of the blood to move out other than the cells. So this is one podocyte that has been shown over here. Only one has been shown, but obviously there is another one that is going around the other side. So let us see that part as well. So here I am drawing actually another podocyte. That is now going around the other side. So there are several podocytes let us draw it here. So which means that now you cannot see, if the podocytes are covering the blood cells, you cannot see the capillary. It is completely enveloped. Try to understand what I am trying to explain to you. So on this diagram, this is the blood vessel. You are able to see the blood vessel, but if I show the podocytes like this on the surface, like wriggles, it means like this yellow part which I am showing in a winding manner. Now it means that you cannot see the glomerulus. The glomerulus is completely enveloping or covering up the entire glomerulus. Why is this happening? Because this is a kind of an additional sieve, additional protection to prevent large proteins and cells from escaping from the blood into the lumen. So this space that you are seeing over here, this is like a slit. So podocyte envelops capillaries in the glomerulus. You think of the blood vessel here like a bamboo pole, and think of your two hands completely encircling it with one set of fingers and other set of fingers into each other, leaving very little space, but that little space is called as a slit. So this is very useful in preventing the movement of blood cells, that is the RBCs, large cells, as well as the large proteins from moving out during ultrafiltration. So from this blood vessel, only the plasma will get into this part. Let us complete that particular diagram. So here was the branch of the renal afferent artery, and what now emerges out will be the renal efferent artery. You can distinctly see that this diameter is less and this diameter is more. Let us continue with this diagram. Here, this will be the luminal space which will continue, but it now continues rather as an extremely convoluted structure, which I will just show like this. It actually has got several loops over here, but I am not showing all the loops, and this continues up to this point. Since this part is closer to the Malpighian tubule, this is proximal, and this is therefore called as proximal convoluted tubule or PCT. Let me erase this part away. So the first part was the Malpighian tubule. The second part is the PCT. Now, if you take a section of the PCT anywhere, we will see that the cells, so see, this is the internal space, this is the lumen. These cells are columnar cells or columnar epithelium. These are called as columnar epithelium, and they have a lot of mitochondria, and they also have microvilli. So this will have microvilli, which we can show over here. And they have a lot of mitochondria, but I won't be able to show it because the size is too small. So I am repeating over here, this is the proximal convoluted tubules in which the cells are tall, columnar, with mitochondria and with microvilli. Now from here, it will continue for some distance and then form a U-tube like structure. Say starting from here. and you can see that this U-tube structure is being formed. This is called as loop of Henle or Henle. So the third part is the loop of Henle. It has a descending limb and an ascending limb. This is very thin, and we will learn about its function after some time when we discuss the function. So where is actually the loop of Henle? Let me point out. So the loop of Henle is somewhere from here. This part is the loop of Henle. See, this much part is the loop of Henle. If the nephron is going right into the medulla, then this part is longer. If it is only restricted to the cortex, then this part is shorter. So the loop of Henle, it is thin tube. It is made up of two parts, a descending and an ascending limb. And the fourth part is the distal convoluted tubule. So as it comes over here, since this is farther away, however, the DCT is coming very close to the Malpighian tubule. So when we use the word distal, it is not only the physical proximity, it is the distance from this end. So you see, this is immediately following. The proximal convoluted tubule is starting immediately from here. Therefore, it is called proximal convoluted tubule. So after some distance, after coiling around, then it comes close to it. Even if it didn't come close to it, it is the distal part, and therefore this part, this is called as the distal convoluted tubule, the DCT. And you can see that it is coming very close to this. This has got a functional significance. It's very important in regulating blood pressure, but that we are not going to discuss now. So this has got a role to play in the distal convolute, in the functioning or in the regulation of blood pressure. Now, just for comparison, this is DCT and this was PCT. The cells are columnar over here with mitochondria and with microvilli. In DCT, the cells are more cuboidal. The cells are shorter, cuboidal, and they have very few mitochondria. And they don't have a microvilli. They don't have microvilli. So they are cuboidal. So this is the difference between the two. And then from here, this part of the DCT will go into the collecting duct. What this means is that this is receiving the distal convoluted tubules from other parts or from other such nephrons together. And do you remember that this is going to lead into the papilla, and then into the minor calyx, and then into the major calyx. So this would be more or less the structure of just the tubule. But we have not considered over here the blood supply. Let us come back to this original diagram. I want you all to consider this and think about it. Here, blood from the renal artery, the branch of the renal artery, is entering into this knot. There is a great deal of pressure over here. Because of the great deal of pressure, most of the blood, but without the cells and without large proteins, is going to be filtered over here. Naturally, this contains many substances that the body requires back. It will have to be collected back. This is going to be done by the blood circulation. So when this renal efferent arteriole emerges out from here, it comes back. Now, it will be little difficult to show it in this double structure, so I will use a highlighter. So instead of using a double line structure, I am only showing this blood vessel with a single thick felt pen like structure. So this is going to go all round, but this is going to branch. Remember that it will go throughout this entire tubule. So this is now branching off and we are showing it in red color, but very quickly that it is getting the characteristics of a vein. This was now a some are going underneath it and some are going above it. I hope you understand this. So this is red color, which means that this is an artery, and very slowly, or an arteriole actually, and it is now re-emerging out and it is becoming or having the characteristics of a vein, because it is slowly becoming deoxygenated, and as it is coming out, it is going to emerge and go off as a branch of the renal vein. And so these are all, this is actually a venule. And this will ultimately go and join as two renal vein. That means it is going to become a renal vein. So you can look at this blood supply to your right side, and what we will say is that the renal efferent arteriole now recapillarizes. That means it capillarizes again. So this is the second bed of capillaries. It recapillarizes again. And what does it do? Where do the capillaries go? All around tubules. Mainly PCT. Mainly into the PCT here. Completely going all around the proximal convoluted tubule. Emerges as, what happens then? It emerges as a venule. You see, this is arteriole and this is venule. All these venules ultimately, they are going to emerge after they join one another as the renal vein. And whatever we have described now, all this that we have described now, is called as peritubular circulation. If this is much deeper and if the nephron is very long, they are also referred to as vasa recta, if they are very long. Otherwise we call this as, or simply you can remember the term peritubular. Why peri? Peri means surrounding, and tubular because it is around the tubule. Okay. And then it goes down as loop finally and then moves up into the collecting ducts. So this was the blood supply that we have now talked about. Let us talk about the actual process or the physiology of formation of urine. The physiology of formation of urine. Let us get back to this diameter. In this part now, here there is a blood that is coming here with a lot of force, and when that blood or that fluid is hitting the walls, naturally there is a lot of pressure created. It breaks up into capillaries, but the walls or the diameter of the renal efferent artery is much lesser. So this creates a greater pressure over here. Because of this very great pressure, the blood is forced down. But when we say blood is forced down, it doesn't mean entire blood. It only means blood but without cells and without proteins. So this is working under high pressure. So the first step is called as ultrafiltration. Why does it happen? Because the forward pressure is greater than the back pressure. The back pressure may be because of, it is in fact because of the osmotic pressure that it is creating. It's also because, as I told you, the diameter of the renal efferent arteriole is a little less, and therefore the forward pressure is greater than the back pressure, and therefore blood without, it is very important to remember this, without cells and proteins. That means most of the plasma, that is plasma, enters lumen of PCT, that is the proximal convoluted tubule, and this is called as preliminary urine, because this is not the same urine that is going to be passed down, and it is good that it doesn't pass down. Let us try to say over here that about 180 liters of fluid or plasma moves into PCT, or you can simply say that 180 liters of blood enters into the kidney. And how much blood do we have? We have just about, say, 5.5 liters of blood. So 5.5 liters of blood, and per day 180 liters of blood is entering into the kidney. If it is going to be all passed out as urine, we won't live even for one day. Which means that a lot of the blood, that is more than 99% of plasma, is returned back. And therefore, this particular piece of information is very relevant to what we are discussing in the next step, which is selective reabsorption. So in the first step, what happened? Ultrafiltration. Forward pressure is greater than back pressure, and therefore almost all the plasma, no cells and no proteins, they enter into the PCT. Okay, they enter into the PCT. But we must remember that a lot of the blood goes into the kidney per day. Therefore, we have to get it back. And getting it back is called as selective reabsorption. So it will be reabsorbed either by active transport or by diffusion, which may happen through special ion channels. Water, of course, moves out through aquaporins. And where is all this located? Let us go back to the diagram over here. In the PCT over here, in the PCT. So if you assume that so we have just drawn over here a part of this. A part of this has been shown over here. So these are the cells of the PCT, and these have got lot of microvilli. This is the luminal space. Now, remember that here I am just showing here for representative purposes blood vessel. This is the blood vessel, this particular blood vessel. So let us use this yellow color to mean that there is a fluid. The preliminary fluid is entering over here into this luminant space, and it will keep going down. But as it does so, whatever the body requires back, it will move across the cell from here to here. By which method? By either active transport, either by using diffusion or by special ion channels. That is why there is so much or so many numbers of mitochondria, because active transport has to be used, and for many of these processes perhaps ATP is required. So this is the second step of selective reabsorption, very crucial and very important. And the third step is tubular secretion. Some substances, to some extent, let's say potassium ions, to some extent substances like creatinine, sometimes drugs which have escaped entering into the lumen, these all are directly secreted into DCT. Many of these substances, they will get directly taken into the tubules of the DCT. So in all these parts over here, from the blood, whatever could not be taken into the glomerulus, and from the glomerulus into the PCT, these substances like ammonia, sometimes a small amount of ammonia or small amount of potassium, sometimes some kinds of drugs, creatinine, all these substances will be directly be pushed from the blood into the distal convoluted tubule. So there are three steps that we have seen over here. The first step takes place in the Bowman's capsule. The second step takes place, that is of reabsorption, takes place in the PCT, and the third step in the DCT, which is of tubular secretion. And this filtrate is going to, primary filtrate is going to go down this, and many substances will be reabsorbed back over here. Salt and water are reabsorbed, so that when the filtrate passes down, so here this preliminary urine can also be called as preliminary filtrate. But this has not yet become urine. It will become urine only when there is more reabsorption of water and ions. So let us look at the next part, that is osmoregulation. Which will take place in the loop of Henle or Henle's loop. As we have seen, there are two limbs to it. One is called as descending limb. Now the descending limb is impervious, does not allow, impervious to Na and Cl, but allows H2O. In comparison, or by contrast, the ascending limb is impervious to H2O, but allows Na and therefore Cl2. So let us get back to that diagram. This is the descending limb, This is the descending limb, this is the ascending limb. Now in this region over here, the amount of salt, this is this part is this region is the interstitial tissue with lot of cells, etc. Now in this interstitial tissue there is an increasing concentration of NaCl. It means it becomes more hypertonic. Okay. So there is more NaCl here and there is very less NaCl there. So from here there is very high concentration of NaCl, whereas as we go up, less concentration of NaCl. So what happens as the result of that? For that we have to think a little bit about what we mean by osmosis. You see, as the filtrate is coming down, whatever concentration of NaCl is outside, because there is a difference, the water from here will emerge out. Why? Because we just said that the descending limb is not going to allow NaCl to move in and out. It is only going to allow water. Because the concentration of NaCl is more outside, water comes out. A simple principle of osmosis. In the next step, again there is more concentration of NaCl here. Again the H2O will come out. Here again NaCl concentration is more over here. Again more H2O will come out. Every step throughout this loop, H2O keeps coming out because there is an increasing concentration of NaCl. This is the beauty of the loop of Anle and of osmoregulation. That means when the urine or the preliminary filtrate is coming over here, it has become highly concentrated. Now when it is emerging back and moving upwards in the ascending limb, we just said that it is not going to allow water, but it is going to allow only NaCl to move. So from here a lot of NaCl moves out. So I will just put it as Na over here because I can't show both. So Na. So a lot of this Na keeps coming up. And when we mentioned here that there is more concentration of NaCl, this comes from here only. This both salt, this NaCl salt as well as little bit of urea, both of this will come over here. And naturally more and more of the NaCl will come out over here, and by the time it comes out, the body has been able to reabsorb both water also and NaCl also. The same thing takes place in the ends of the collecting ducts here also. Here also water emerges out. So we will write this down. We have mentioned here, recollect that what we have said, descending limb impervious to Na and Cl but allows water. Ascending limb impervious to H2O but allows NaCl. So in other words, the ascending limb controls the descending limb, because in the ascending limb NaCl is allowed to go out. This NaCl, when it goes out, it pulls the water out. So this is called as counter current loop and counter current loop flows. Studied a lot in engineering as well. You can try to find out more about counter current loop principles, where all in the body such countercurrent loop mechanisms they flow. So this is by a method of countercurrent loop mechanism. That is why there is a loop, and that is how this structure has evolved, and this has been adapted because it is very successful. In what happens is that prevents loss of water and salts. It means NaNCl, and this is under the control of antidiuretic hormone. So regulated by antidiuretic hormone. But the scope of this is not there in the syllabus, how it happens, etc. We just mentioned that the ADH is there. Similarly, we will just say that the renin and angiotensin mechanism to regulate BP or blood pressure is very important. Renin angiotensin mechanism. Once again, that is not in the scope of the syllabus. We have to just learn about structure and function. So we have learnt over here how a miraculous structure and organ like the kidney is able to control our body in so many ways. It's one of the most important organs in the body, and therefore be kind to the kidney and don't pressurize it, because the kidney is so very important for a healthy lifestyle. That is the note that I wanted to end this lecture with. So thank you, and Professor Ramindranath signing off.